Welcome back everyone. Today we are going to start the discussion of this nerve muscle physiology. In this nerve muscle physiology sub part of physiology, we are going to start the discussion of characteristics of nerve fibers. We know nerve fibers. Okay, so let's start with it. Okay, now we all know from the basics that the functional unit of nervous system. Our nervous system's functional unit is neurons or nerve cells. Okay, neurons or nerve cells. These are the functional units of our nervous system. Okay, so with this basic, let's start the first basic topic: cells in our central nervous system. Cells in CNS. Okay, in cells in CNS we have two types of cells. First is our neurons, obviously, and the second types of cells are called glial cells. Glial cells. Remember, the ratio here of neuron and glial cells is 1:10. Okay, that is, glial cells are more in number. More than neurons, there are glial cells. The ratio is 1:10. Okay, so let's start with the discussion of glial cells first. Okay, glial cells. Okay, these glial cells, we have two types. We have two types of glial cells. Okay, first is the bigger glial cells, macroglial cells. Macroglial cells. Okay, macroglial cells. And the other one is the microglial cells, the smaller ones. Bigger ones macro, smaller glial cells are micro. Okay, the macroglial cells, we have the star-shaped cells called astrocytes. Okay, then we have oligodendrocytes. And then we have Schwann cells. All of their functions we will learn afterwards. Oligodendrocytes and Schwann, SCHWANN, Schwann cells. Now in the microglial cells, okay, these are different in terms of embryological origin. That is blood monocyte of macrophage lineage. Okay, these are different. Microglial cells are different in terms of their embryological origins. For example, in blood monocyte, these are macrophage lineage. Okay, now let's start with the discussion of our macroglia. First we have astrocytes. Okay, so astrocytes, these are the star-shaped cells. The star-shaped cells in our central nervous system. Okay, these are also of two important types. First is a protoplasmic astrocytes. Okay, these are present only in gray matter. Okay, the other one is fibrous astrocyte. Protoplasmic astrocyte only in gray matter. Fibrous astrocyte in white matter. Okay, gray matter protoplasmic, white matter fibrous. Okay, now an important fact here: there are the intermediate filaments in astrocytes. Intermediate filaments in astrocytes. Okay, these are composed of GFAP. Okay, these are glial fibrillary acidic protein. Glial fibrillary acidic protein. This, remember, this GFAP is a tumor marker for astromyotas, astrocytomas, carcinoma of the astrocytes, so astrocytomas. The tumor marker is this GFAP, glial fibrillary acidic protein. These are intermediate filaments in the astrocytes. Okay, astrocytes are of two types: protoplasmic and fibrous. Protoplasmic in gray and fibrous in white. Okay, now the functions of astrocytes, very important, commonly asked functions. First function is our triple B. You know that this is the blood brain barrier. Okay, so how this blood brain barrier is formed, we are going to see now. So this is the blood vessel. Okay, vessel containing the blood flow. Okay, now these are the what, endothelial cells. Endothelial cells in the blood vessels. Okay, these endothelial cells composed of, remember, tight junctions of endothelial cells. Okay, now this is the brain tissue. Okay, this is the brain tissue here. This is the brain tissue. Now here we are going to draw the astrocytes, these star-shaped cells. Okay, now these astrocytes have something called foot processes. Okay, these are the foot processes. Okay, foot processes of astrocytes. Okay, these are what, foot processes of astrocytes. This is brain matter. Okay, now this blood brain barrier right here, BBB, blood brain barrier, is due to, first is the foot processes of astrocytes, and the other one is the tight junctions, of endothelial cells. Foot process of astrocytes and tight junctions of endothelial cells contributes to blood-brain barrier. This was the function of astrocytes and blood-brain barrier. Then next function is synaptic cleansing or clearing or cleaning. Synaptic cleansing. So there are signals. So following signals. Okay, there is uptake of potassium neurotransmitters into astrocytes. Potassium and neurotransmitters into astrocytes is taken up following signals. These are very important for effective functions of neurons. Synaptic cleansing. Okay, so these are two most important functions of astrocytes: formation of blood-brain barrier or contribution of blood-brain barrier, and synaptic cleansing. Okay, so first macroglial cell, astrocytes, is over. Okay, next we're going to see is our oligodendrocytes. Okay, yeah. So we are going to discuss the two cells, so oligodendrocytes and Schwann cells together. Okay, why we'll see later. Now these two cells in the CNS, these two, the combined works of myelination. We know the myelin sheath around the neurons. This is done in the central nervous system, both by oligodendrocytes and Schwann cells. Where in the CNS? Now this one differs. Okay, we are going to see where in the CNS. The oligodendrocytes do in myelination in CNS fibers. Okay, the Schwann cells do the myelination in PNS fibers, peripheral nervous system fibers. Oligodendrocytes in CNS, Schwann cells in PNS. That's the job of myelination. Okay. Now, oligodendrocytes in CNS is capable of multiple myelination. One of the features of oligodendrocytes in CNS. Now, Schwann cells in PNS capable of single myelination. What do you mean by this? A single oligodendrocyte cell can myelinate multiple neurons. Okay, but here the single Schwann cell will myelinate only a single peripheral neuron. It is the ratio here is 1:1. Okay, here the ratio may be nearly 1:4 or 1:3, something like that. Okay, but generally it's 1:4. Here the ratio is 1:1. Single myelination Schwann cells in PNS, multiple myelination in CNS by oligodendrocytes. Okay, both oligodendrocytes and Schwann cells, the function of myelination. This was all about our microglial cells and their important functions. Now, moving towards our important discussion, that is of our all important neurons. Okay. Now I am going to start the discussion of neurons. So here we will start by seeing the types of neurons. Very simple discussion, types of neurons. Okay. First is our unipolar neurons. Unipolar neurons. Okay. How's the structure? This is a single cell body and a single axon. Okay, these are unipolar neurons present in invertebrates. These invertebrates are lesser developed organisms than the vertebrates and other phylum of organisms, so they have unipolar neurons. These neurons are not much developed, so these are in invertebrates. Unipolar neurons in invertebrates. Okay. Next, we have our pseudo unipolar neurons. Okay, pseudo unipolar neurons. Now, the pseudo unipolar neurons, why this is called pseudo unipolar? Because it has a single cell body, okay, no dendrites, a single axon for transmission of signal, but this axon divides into two parts. Okay, this is a pseudo unipolar neuron. Okay, now this division, one division goes, for example, goes to skin. Okay, another division goes to spinal cord, into skin, into spinal cord. So from skin it fetches signals, and through these neurons by processing sends this signal through this division to the spinal cord. This pseudounipolar neurons is very important. It is present in dorsal root ganglion of spinal cord. Pseudounipolar neurons in dorsal root ganglion of spinal cord. Okay, one axon divided into two divisions or functions. The third type of neuron, very common, are bipolar neurons. Okay, as we say bipolar, so it must have a single cell body, a dendrite for receiving information, and an axon. So bipolar, two poles for receiving and transmitting information. Okay, dendrite to receive information, axon to transmit information. Very basic understanding, we know everything from before, just completion sake we are discussing. These are present in, remember, first is retina, or all important retina. Okay, the other one is, remember, olfactory epithelium, the nose, very important. Retina in the eyes and olfactory epithelium in the nose. These bipolar neurons are present. Okay, as a dendrite to receive information and an axon to transmit information. These are bipolar neurons. And then finally we have multipolar neurons. Multipolar neurons. Okay, so as we have known from the spelling, we will have multiple dendrites for receiving information, but a single axon to transmit. Okay, these multipolar neurons are the most numerous ones, and they are present in spinal and motor neurons. Spinal and motor neurons are the greatest example of multipolar neurons, which are the most numerous ones in our body. Okay, bipolar neurons in retina and olfactory epithelium. It's simple and easy to remember. Remember olfactory epithelium. What an example. We know all this example of retina, but olfactory epithelium is a new one. Okay, pseudonuclear and dorsal root ganglion is what's a special functional value for transmission of sympathetic parasympathetic impulses in the spinal cord, and then there in the invertebrates is a unipolar neuron. Okay, less developed neuron. Now with this types of neuron discussion, we have moved to structure of neuron. Structure of which neuron we are going to discuss is structure of, remember, multipolar neuron. Okay, so let's start with a simple diagram. Okay, we are drawing a cell body. Okay, say this cell body, we have these components. Okay, now the cell body will obviously have an axon. Okay, along with this axon it will have some dendrites. Okay, we will draw some dendrites for receiving information, and the axon is for transmitting information. Okay, now this axon will have some cells. You have to see everything one by one. These are the cells covering the axon. The cells are for myelination. You know what they are. Okay, so let's name one by one. First, okay, first this is the, we know, cell body, or also called soma. Okay, and this is the obviously nucleus of the cell body. Nucleus of cell body or soma. Okay, now these are the dendrites. These are the dendrites. Now the structures in the cell body. What are these? These are, remember, Nissel's granules. Nissel's granules. These are, remember, rough endoplasmic reticulums. What rough endoplasmic reticulums do? They have ribosomes for obviously protein synthesis. So Nissel's granules are for protein synthesis. Why this protein synthesis is required? For survival of neurons. Very simple. For survival of neurons, proteins are required which are synthesized by Nissel's granules in the soma. What are these Nissel's granules? Rough endoplasmic reticulum containing ribosomes. Okay, clear? Now moving on from the cell body, completed the cell body, to a discussion on the axon. Okay, this whole total green of this is called as axon. Very simple, we know. Remember, this axon has some parts. So this part of axon is called, remember, initial segment of axon. Initial segment of axon. Okay, above this initial segment of axon we have this part, this the junction between the initiation of axon and the cell body. This is called, remember, axon hillock. Axon hillock. Let me write this again. Hillock, HILLOCK, axon hillock. This gives rise to axon. Gives rise to axon. Below this axon hillock we have the initial segment of axon. Now this axon is for conduction of impulses. Yeah, very simple, conduction of impulses. Okay. Now what are these cells in violet color? To discuss these cells, these are called myelin. These are myelin. Okay, these are myelin. Now we see that this presence of myelin, let me extend this. You see, this presence of myelin is discontinuous. Here and here and here, this myelin is discontinuous. Okay, no myelin. Okay, these are called nodes of Ranvier. Nodes of Ranvier. Okay, here speciality of nodes of Ranvier are the concentration of sodium channels are maximum in nodes of Ranvier. Concentration of sodium channels are maximum in the nodes of Ranvier. The concentration is, remember, 2000 to 12,000 per square meter. This is the concentration of sodium channels which is maximum in the nodes of Ranvier. Okay. Now here this axon is for conduction of impulse and this is myelin. Okay. Now this conduction of his impulse is two types. It is either fast or slow. Okay, fast if myelin is present and slow if myelin is absent. So one of the concepts to remember here, myelin is present, the conduction is fast. How we'll see later. Myelin is absent, the conduction is very slow. Okay, so this was the ultrastructure of a neuron. There is different parts. Okay, now at the end here, the end of this axon, we have important synapse. It's the last part. At the end of the axon we have a synapse called synaptic terminal. Okay, synapse with synaptic terminal at the end of the axon. Okay, now I'm going to see some conduction types in the neuron. Okay, the conduction is from cell body to synapse through axon. Okay, cell body to synapse through axon. This is called anterograde conduction. Anterograde conduction. This is with the help of kinesin protein. Remember the intermediate filaments, kinesin, with the help of kinesin protein. Kinesin with help of anterograde conduction. This can be fast or slow due to presence or absence of myelin. Okay. Now the conduction can be in the opposite direction, that is from synapse to the cell body, in the opposite direction through the axon. This is important, so I'm starting marks from synapse towards the cell body through the axon. Okay, now this is called reverse axonal transport or retrograde conduction in the opposite direction, that is why retrograde conduction. Here it is due to kinesin, here is due to dynein protein. With the help of dynein protein, dynein, dynein protein. It's one of the intermediate filament proteins, kinesin and dynein. If you don't know, check out the intermediate filaments video on this channel with the help of dynein protein. Now this reverse axonal transport or retrograde conduction with the help of dynein protein is used for virus invade, or infection. Various virus invade and infection, they use this retrograde conduction method with the help of dynein protein. Okay, these was two types of conduction and their importance. Okay, now this protein, kinesin and dynein protein. Okay, next moving on, I want to discuss the all important action potential generation. Action potential generation. Okay, action potential generation. Now action potential generation is different in different types of neurons. Okay, in motor neurons, okay, in the sensory and motor neurons. Okay, motor neurons, it is initiated from the initial segment of axon plus axon hillock. Okay, we have seen this axon hillock and initial segment of the axon here. The axon hillock and initial segment of axon. In the motor neurons, the action potential generates from here, and in sensory neurons, initiated from, remember, the first node of Ranvier. We have seen before, the nodes of Ranvier are the highest concentration of sodium plus channels. So the first node of Ranvier is the site of generation of action potential in sensory neurons. In motor neurons, axon hillock along with initial segment of axon. Okay, moving forward, we have due to presence of myelin, okay, or absence of myelin, If myelin is absent, this conduction is slow. If myelin is present, this conduction is fast. Okay, this myelin has the function of insulation and increased thickness. This myelin has the function of insulation and increased thickness. It increases the thickness of the axon. Okay, now this myelin is composed of lipid and proteins. Composed of lipids and proteins. Now which lipids and proteins we are going to see here. Now lipids is 80%, proteins is 20%. Okay, what are the lipids and proteins we are going to see here. The lipids is sphingomyelin. Obviously for myelin we have our sphingomyelin, and the proteins are called zero proteins or myelin basic proteins. Myelin basic proteins, MBPs. Myelin basic proteins of the lipid is sphingomyelin. Okay, now this myelin basic protein, the zero protein, this characteristic feature here is this is easily damaged by autoantibodies. Easily damaged by autoantibodies. Okay, so this leads to obviously demyelination. And obviously decreased nerve conduction velocity. Nerve conduction velocity, in short NCV. Okay, they are easily damaged by autoantibodies, which zero proteins or myelin basic proteins. This leads to demyelination and decreased nerve conduction velocity. Okay, this leads to, remember, this condition called multiple sclerosis. Multiple sclerosis. Okay, so multiple sclerosis is due to damage of these zero or myelin basic proteins by autoantibodies, leading to demyelination and decreased nerve conduction velocities. Remember, multiple sclerosis. Okay, very important clinical feature. Moving on, as we are discussing the nerve conduction velocity, it's important to discuss here the factors affecting nerve conduction velocity, NCV. Okay, see the factors. First factor is diameter. Okay, is proportional to nerve conduction velocity. It is a linear relation. Okay, diameter is proportional to nerve conduction velocity. The next factor is obviously myelin. You know, myelin increases nerve conduction velocity. Presence decreases nerve conduction velocity. Absence. Very simple. Okay, why? Presence of myelin increases nerve conduction velocity. We'll see after this. Third point is thickness. Okay, you seen above, this myelin causes insulation and increased thickness. This thickness factor is proportional to nerve conduction velocity. Okay, this thickness is due to myelin. Okay, now this thickness or myelin, increased thickness, remember, decreased, it causes decreases membrane capacitance. Decrease membrane capacitance, so increasing nerve conduction velocity. What do you mean by membrane capacitance? The capacitance concept is the ability to hold the ions or charges. So as the charges will be quickly removed from the membrane or the axon, the faster will be the conduction of nerve signal. Okay. So as decreasing membrane capacitance will increasing the nerve conduction velocity. Okay. Now here from this we know that thickness is inversely proportional to membrane capacitance. Membrane capacitance. Capacitance is the ability to hold charges or stored charges. So here another discussion we're going to have how this presence of myelin increases the nerve conduction velocity. Okay. Here there will be, due to presence of myelin, increased thickness. This increased thickness will lead to axonal resistance. Axonal resistance. Okay. Now resistance is what? It's a resistance to the flow of charges in the conduction of impulses. Impulses are conducted by charges. Capacitance is storage. Resistance is stoppage. Okay. This will increase the axonal resistance. So there will be no diffusing out of the ions. Okay. This is the axon. This is the myelin layer that is developed. This will not allow the diffusing out of the ions. The ions will not diffuse out of here. Okay. So instead of diffusing, it will directly go to next node of Brandwehr. Okay. This is one of the factors favoring nerve conduction velocity. No diffusing out of ions due to increased axonal resistance. Important point to remember. Due to increased thickness due to myelin. Okay. The fourth factor here is increased temperature. Increased temperature will increase nerve conduction velocity. Okay. Decreased temperature, nerve conduction velocity is delayed. Yeah, these are the four factors: temperature, myelin and thickness, and diameter. Okay. The concept of capacitance and resistance. Now I'm going to see the electrical event of the neuron, it is action potential. Electrical event of neuron, that is action potential. Before we go through this, first point we are going to remember is the resting membrane potential of a neuron. Okay, resting membrane potential of a neuron, which is minus 70 millivolt. Okay. Okay. So with this, first of all, we are going to draw a graph. Okay. We are going to draw a graph here. Y-axis, the x-axis, on the x we have time in seconds, and on y-axis we have membrane potential. Membrane potential, which is in obviously millivolts. Okay. Now the resting memory potential of a neuron is minus 70 millivolt. So here the resting memory potential is minus 70 millivolt. Now we give here the stimulus. Here in the time graph we give the stimulus. Okay, we give the stimulus. Stimulus is given here, but the stimulus will not act as soon as the stimulus is given. It will have a time period of lag. Okay, this is time period of lag. Now here after some time, from minus 70 millivolt, the first phase, okay, will shoot up to how much? Minus 55 millivolt. Okay, we'll come back later to this minus 55 millivolt. So this is the first phase. Okay, now after minus 55 millivolt is achieved, this will shoot to plus 35 millivolt, including crossing the 0 millivolt barrier, plus 35 millivolt. So second phase. Okay, after reaching the 35 millivolt and diffusing out of the ions, we'll see what the ions are involved. The third phase comes. What happens in third phase? This happens. Okay, this happens, and to how much it goes? To minus 90 millivolts. Minus 90 millivolts. Okay, this is the third phase, and this is a in dotted lines fourth phase. Okay, the first phase from minus 70 to minus 55. Once minus 55 is reached, the second phase starts crossing the 0 millivolt to plus 35 millivolt. Reaching plus 35, goes back to minus 90 millivolt, crossing the minus 70 millivolt barrier, and then from the minus 90 millivolt slowly getting up back, which is in the fourth phase. Okay, now this stimulus does not act outside, it does not act as soon as it is given. So this is our lag period. Lag period. Okay, called latent period. So the generation of action potential in a neuron has a latent or lag period. Okay, now we'll see one by one the four phases, all important four phases of the electrical event, the reaction potential in the neurons. Okay, so we'll start. The first event, I have did this in blue color. So the first event is in blue color. The first event here is called local potential change. Local potential change. This changes from minus 70 millivolt, obviously the resting event potential, to minus 55 millivolt. Okay, this is solely, this first phase is solely due to, obviously the negativity decreases. Okay, so there is influx of a positive ion, which is our sodium ions. So here there is influx, this is inside influx of sodium, influx of a positive sodium ion, decrease of negativity from minus 75 to minus 55, called local potential change, the first phase. Okay, now as soon as the minus 55 millivolt is reached, there is this phase of all important depolarization. Okay, this depolarization further decrease in negativity. Also this is obviously due to influx of a positive ion, due to influx of positive sodium ions. It's also due to sodium ions. So we'll do this inside influx of sodium. Okay, now this sodium ions influx is through voltage gated sodium ions channels. Voltage gated sodium ion channels. Voltage gated sodium ion channels. Okay, and there is decrease in inside of negativity. So this goes from minus 55 millivolt to plus 35 millivolt, breaching the barrier of 0 millivolt. Okay, now as soon as this depolation event is completed, comes the third phase. The third phase is our repolarization, reversing back to the original polarity, repolarization. And what happens in this repolarization, as we see from this graph, from plus 35 millivolts it reaches the 0 millivolt barrier and reaches the minus 90 millivolt. So the inside the negativity of the cell increases. Inside of the cell negativity increases. So increase in negativity where inside the cell. Inside the cell. Okay. Why due to this repolarization? Remember, due to obviously increase in inside negativity. So obviously a positive ion will be removed or a negative ion will enter. Okay. So this will be due to, remember, this is a repolarization is a universal process. This will obviously due to influx of positive sodium ions. Okay, which is due to slow closure of potassium ion channels. Due to slow closure of the potassium ion channels, there is continued potassium influx, which will lead to increase in negativity of, sorry, not potassium influx, it is efflux, removal. Okay, flux due to slow closure of potassium ion channels. I've also said this increase in negativity could be also due to moving inside of a negative ion channel. So there will be chloride ion influx. Repolarization is due to potassium efflux, removal of a positive ion and inside moving of a negative ion. Okay. In this repolarization phase, these all processes, okay, will lead to our fourth phase, hyperpolarization or hyperpolarization. This dotted phase is our hyperpolarization. The hyperpolarization will be due to all these factors: slow closure of the potassium channels leading to potassium efflux and chloride influx. Okay, hyperpolarization. Now the all important facts here to remember about hyperpolarization. Okay, hyperpolarization is the phase of neuronal function inhibition. There will be inhibition of neuronal functions in this hyperpolarization phase. This will be by inhibitory neurotransmitter. And one of the inhibitory neurotransmitters we know is GABA, that is gamma amino butyric acid. Gamma amino butyric acid. This hyperpolarization phase of the neurons and action potential of the neurons is the phase of inhibition of neuronal function. This is done by inhibitory neurotransmitter GABA, gamma amino butyric acid. This GABA, remember, acts at hyperpolarization phase. hyperpolarization phase of action potential. Hyperpolarization, remember, hyperpolarization is the inhibitory phase which inhibits the normal neuronal functions of the neurons. Okay, now this GABA agonists. What do you mean by agonists? Similarly acting chemical compounds or molecules. So this similarly acting chemical compounds or molecules of GABA, these are used for sedation. Sedation, losing of the body functions and consciousness, as this is the inhibitory phase of the neurons, so the overall body function does not occur in sedation. Now what are these GABA agonists? Remember, barbiturates and benzodiazepines. Okay, for sedation, GABA agonists are used. So barbiturates and benzodiazepines are used. They act at hyperpolarization phase of action potential, leading to inhibition of the neuronal functions. So this is all about the electrical event of the neuron. It is our action potential. Okay. First phase, local potential change from minus 70 resting membrane potential to minus 55 millivolt. Now I've said to remember this minus 55 millivolt. Okay. The time has come to say what is this importance. This, remember, this minus 55 millivolt here is the firing. This is the firing potential. Okay. Firing potential of neuron. Okay. Once this minus 55 millivolt is reached from minus 70 millivolt in the local potential change, action potential fires here. Then in the second phase, there is depolarization from minus 55 millivolt crossing the 0 millivolt barrier to plus 35 millivolt. Local potential change is due to influx of sodium ions. This depolarization phase is also due to influx of sodium ions through voltage gated sodium channels. Then there is repolarization phase, back to the polarity of the neuron, which is due to potassium efflux. Universal repolarization is due to potassium efflux. And upon reaching this phase, there is this depolarization phase, which is due to continued potassium efflux and also chloride influx, increasing negativity inside the cell, which will lead to hyperpolarization phase. This hyperpolarization phase is the inhibitory phase. It inhibits the neural function. The GABA is a inhibitory neurotransmitter. It acts on this hyperpolarization phase of this action potential. So these GABA agonists are used for sedation, such as benzodiazepines and barbiturates. Okay. Now next, in this discussion of action potentials, okay, we want to see the concept of absolute refractory period and relative refractory period. Absolute refractory period and relative refractory period. Very important concept in this action potential discussion. We'll understand this very clearly through a graph. Okay. This is the y-axis, this is the x-axis. Okay. I will draw the action potential graph here. So this was the local potential change, the depolarization phase, the repolarization phase, and hyperpolarization phase. Okay, this was all the phases here. And we draw this demarcation here. Okay, these are the three demarcations here giving two phases to us. Okay, so this first phase, this phase, sorry, not this one, this phase. Okay, this is the absolute refractory period, ARP or ARP. Okay, this consists of firing level minus 50 millivolt firing level along with 1/3 of repolarization, This consists of firing level along with 1/3 of repolarization. Okay, this is absolute refractory period. In this absolute refractory period, remember the voltage gated sodium channels are closed or inactivated. The voltage gated sodium plus channels are closed or inactivated. We are in absolute refractory period. Okay, which consists of firing level, remember this part, along with 1/3 of our repolarization. Okay, now this part is our relative refractory period. This part of the graph, some part of repolarization along with the whole of depolarization phase. So we're going to write this relative refractory period. Okay, this consists of one-third of repolarization along with end of action potential. Next, 1/3 of repolarization along with end of the action potential. After this phase, relative refractory period. Okay, here in this relative refractory period, remember the voltage gated sodium channels are open or at reset. Sodium gated voltage channels are open, contrast to the sodium gated sodium channels are closed in absolute refractory period. Due to this, when we give a second stimulus of maximum strength, this is capable of producing action potential. In this relative refractory period, when you give a second stimulus of maximum strength, it is capable of producing action potential. So this was about absolute and relative refractory periods. Remember this graph. From this graph, you can deduce everything. Okay, these are the two demarcation phases we have done here. Absolute refractory period, 1/3 of repolarization along with the firing level, and the next 1/3 of repolarization along with the end of action potential is relative. Absolute refractory, voltage gated sodium channels are closed, and here it's relative, it's open. Okay, so this is all about our today's discussion on neuromuscular physiology. First, we have discussed about the cells, different types of cells in the CNS and PNS, macroglial and microglial. Then I've discussed the type of neurons, the functions of the glial cells, the action potential phase, the electrical event, and what are the ions involved, influx and efflux of various types of ions, then the all important absolute and relative refractory periods. Okay, an important basic understanding of this topic. Do revise them, guys. Thanks for watching. I will catch you in the next one. Good luck.